Hi, I'm Bill Rapisi, Executive Director of Lymphatic Education and Research Network, and I just want to take this opportunity to thank all of you for joining us today for our Symposium series. I also want to take this opportunity to thank all of you who become members of LE&RN and help support this series so that we can continue bringing this kind of groundbreaking information to you in your homes and offices. I would also suggest for all of you who have not become members of LE&RN, $5 a month, you can become a member of LE&RN and help support this programming and the research that we do. Thanks very much for joining us, and I look forward to having you become a member.
My name is Laura Santambrogio. I'm a professor of pathology, immunology, and microbiology at Albert Einstein College of Medicine in New York City. And my laboratory has been involved for quite some time in the analysis of the lymphatic fluid under different physiological and pathological conditions. And so today I would like to give an overview of the field in the lymph and what my laboratory is also involved. So the blood that circulates through the blood capillaries, it's not in direct contact with the cellular layer of each parenchymal organ. And so tissue, so lipids, protein, small molecule, and nutrients need to be transported from the intravascular compartment through the extravascular compartment in order to provide cellular nutrients and hydration to the cells. Most of this protein will exit the capillaries through pressure-mediated ultrafiltration and will collect here into the interstitial fluid, so they are available to all parenchymal cells. These are the nutrients which cellular in different organs and parenchymal needs. And many of using actually molecular tracer, it has been estimated that around 3 to 6 liters of interstitial fluid are formed every day, depending on the fluid, on the body fluid intake and the general level of hydration. A portion of this protein and interstitial fluid will return to the blood circulation by reabsorption into the venule end of the capillary bed. However, the majority of this product will collect together with a product of tissue metabolism and catabolism and apoptotic cells, and it will be collected into the lymphatic capillaries. These capillaries are open-ended, as you can see, and they open directly among the cellular layer of each parenchymal organs. And so very easily they can collect a large volume of interstitial fluid, apoptotic cells, product of cellular metabolism, catabolism, and upon entering the lymphatic collectors, the fluid will then be called lymph. A very fundamental question is, why do we need lymph? And in general, why do we need lymphatic circulation? So why all the proteins and fluids which are collected into the interstitial space, they do not directly get drained back into the venule, but instead they enter the lymphatic circulation. So why do we need a lymphatic circulation? Physiologically, there are very important reasons why the interstitial fluid does not drain directly into the venule, but will enter a lymphatic vessels. One of the very important reasons is that the blood, the fluid volume in the blood, protein concentration, osmolarity, electrolytes, and pH are very tightly controlled. And so any changes in any of these parameters would have really severe consequences on the body homeostasis. For example, it would be very harmful if the blood pH would change following tissue acidosis, which is normally observed during an inflammatory event, or if all the lipids which are absorbed through the intestine after a heavy meal would be delivered directly into the bloodstream. And that's why the blood homeostasis needs to be tightly controlled, and so it cannot adjust to these changes in volume, in pH, in protein concentration, without really being very harmful to the blood homeostasis. On the other hand, the lymph can sustain a much wider changes and variability without compromising the body homeostasis. You can see how the composition of the lymph can really change. This is physiological lymph. This is after a meal, and you can see just by the color how dramatically the lipid concentration change in the lymph. Or even more so during infection or during a trauma. You can see that there are changes in the biochemical and cellular composition on the lymph. And yet the lymph can sustain this wide range of variability without compromising the body homeostasis. So altogether, we can say that the lymphatic fluid acts as a buffer for the metabolic and catabolic changes in every parenchymal organ as observed during physiological and pathological condition, and can sustain these changes in its biochemical composition without really compromising the body homeostasis. So this is one of the very important reasons why all this product of tissue metabolism and catabolism can be easily drained into the lymphatic circulation, but not directly into the bloodstream. A second important reason is that the lymph, upon collection in all peripheral tissue and all organs, will be transported to one of the 500 to 600 lymph nodes that are present in the human body. And so the nodal cells will be constantly exposed to the self and non-self proteome, which is incoming from each parenchymal organ. And this really ensures that the immune system is aware of what's going on in peripheral tissue, and invading pathogen, for example, can be seen by the nodal cells, but and captured by these immune cells which are present in the lymph node, but they do not enter directly into the bloodstream. So overall, these are the two major reasons why do we need a lymphatic circulation. One is to buffer all these changes in the metabolic, catabolic activity present in each parenchymal organ, and so buffering them and transporting to a lymph node before entering the blood circulation. And the second is just to be aware of what's going on in every parenchymal organ, to be able to capture any pathogen that enter peripheral tissue. And also to be aware of the self proteome that it's constantly presented by the lymphatic circulation to the immune cells that are present in the lymph node. This concept that the lymph directly capture the organ proteome and the product of tissue metabolism and catabolism has been developed through the years. Several laboratories have performed proteomic analysis on bovine, ovine, and rodent lymph, and my lab was the first one to perform the proteomic analysis on the human lymph. And all these analyses have indicated how the lymph composition does really not overlap with the one of the plasma for the reason that I mentioned before, and that the lymph composition is really much more enriched of proteins that comes from different parenchymal organs. This is just one experiment that shows how different labs have been mapped all these protein coming and being present in the lymph. For example, this is a 2D Dij analysis in which lymph protein are labeled in green, plasma protein are labeled in red, and so all the protein that are present in both biological fluids will be seen in yellow, whereas protein which are only present in one of the two biological fluids are the green or red, and we can pick these spots and analyze it by mass spectrometry. Other more comprehensive analyses rely on 1D gel analysis in which you take the lymph, for example, of different individuals. Here are 9 different people. We took lymph or blood. Sometimes it's very hard to take enough amount to load on the gel, so we need to pull the lymph or the plasma from different people. Then we run on the gel, and then each protein, this part of the gel, are analyzed by mass spectrometry. And you can see that each one of these peaks correspond to a fragment of a certain protein, and so you can see how much the lymph fluid is enriched in different proteins. And finally, this is a recently performed metabolomic analysis, also indicate the complexity of the metabolic pathways which are present in the lymph. Here is just one of the many examples. It shows the arachidonic acid prostaglandin pathway, and many of these metabolites have been found in the lymph. And so altogether, these analyses have indicated that in comparison to the plasma, the lymph is really enriched in products that derive from cellular secretion, apoptosis, extracellular tissue remodeling, and in general, tissue metabolism. Importantly, not only the lymph proteomic, it's different from the one of the plasma, does not overlap with the one of the plasma, but the lymphatic fluid also show an organ tissue specific signature. So the lymph that is collected from different lymphatic districts, for example, the pancreas, the lung, the intestine, really reflects the protein composition of that particular organ. And more importantly, during different inflammatory or infective conditions, tissue-specific antigen will increase very early on in the lymph as compared to the plasma. And a few labs, for example, have calculated how tissue-specific antigen are between 500 and 1,000 times more concentrated in the lymph as compared to the blood. And this is very important because it indicates how the lymph could be a biological fluid to be used for biomarker discovery. So, for example, very early on, discover of a sign of cancer in a particular organ. And the lymph that will be collected from all these different parenchymal organs, and that, as I mentioned a few minutes ago, contains the molecular signature, the protein signature of each one of these organs, will then be collected into the lymphatic and transported to the lymph node. I'm just going to show a movie now that was taken by Anatoly Gashev at Texas A&M. In this movie, you will see the blood circulation. See, here is a blood capillaries, and you can see all the red blood cells that are moving very fast. And this is a lymphatic collectors. And we can measure the strength and the frequency of the contraction. You can see how the lymphatic collector is contracting and transporting the lymph to the lymph node. The lymphatic collector has an intrinsic pumping activity. You could see in the movie how the lymphatic was pumping the lymph to the lymph node. And the reason why there is this pumping activity is because there are some muscle cells into the collector in the outside part of the collector that will function as an intrinsic pacemaker. It's very similar to the one in the heart. They can spontaneously depolarize, and through a slow current of calcium, they can initiate the vessel contraction. We don't know much yet about these muscle cells with this pacemaker activity, but a proteomic analysis that we performed recently in the lab, in which we isolated the collector and then we basically analyze all the proteins that are present in these lymphatic collectors. And we confirm what previously thought, that these lymphatic muscle cells have characteristics which are similar to smooth muscle but also to striatal muscle. And we also map several of the sodium, potassium, and calcium ions channel that allow membrane depolarization and muscle contraction. So in the future, it would be very interesting to learn more about these lymphatic muscle cells. Since in the US alone, there are over 10 million people with secondary lymphedema that could be ameliorated by increasing the lymphatic pumping activity. And so it would be important to learn more about the activity of these muscle cells, which are the ion channels, which are important for maintaining the pumping activity of the collectors. Through this pumping activity and a series of valves which are positioned inside the vessel to prevent backflow of the lymphatic fluid, the lymph is then transported to the draining node. Here you can see a draining node that has been collected. So this is a lymph node, and here is a part of the intestine. And you can see all these lymphatic collectors that will transport the lymph to the lymph node, which is inside at the center of these slides. Once the lymph enters the lymph node, it can travel through two different pathways. One is the lymph will enter the afferent lymphatics and then will travel in the subcapsular sinuses. These are lacunae which will be full of lymph. And in these lacunae you have several macrophages and dendritic cells, which are immune cells, which are positioned at this level because they will take any incoming pathogen, any bacteria, any viruses that enter through the lymph. It will be phagocytosed by these antigen presenting cells. The lymph then can either from the subcapsular sinuses go to the medullary sinuses and then exit through the efferent lymphatic and will go to another lymph node. Or it can enter from the subcapsular space, enter the conduit system, and the conduit system opens directly into the bloodstream through the high endothelial venue. So any product that will enter the conduit system can then go into the blood circulation. And there is an anatomical barrier between the conduit system and the subcortical space, which is around 70 to 80 kilodalton. And so any proteins or any product which is higher in molecular weight at 70-80 kilodalton will not be able to enter the conduit system, but will follow this pathway: subcortical sinuses, medullary sinuses, and then exiting through the next to the next lymph node. And this is a very important protective mechanism because it ensures that pathogen, bacteria, viruses that are not trapped by the macrophages and the dendritic cells into the subcortical space, they can be delivered then to the medullary sinuses, which are very similar to the subcortical sinuses. They are also very enriched in dendritic cells and macrophages. But if they are not phagocytosed, even here, they will go to the next lymph node. And so there is another chance for the immune system to capture this pathogen. Whereas it would be very harmful if the pathogen can directly enter the bloodstream. And that's why we have this protective mechanism, this barrier that makes sure that pathogen can only follow this route. And only very small product below 70-80 kilodalton can directly be transferred from the lymph into the high endothelial venue. Overall, there are several evidence that the lymph node collect from specific area of the body, and they really can capture the incoming proteome from each parenchymal organs. For example, injection of fluorescent tracer as indicated, like if we inject the fluorescent tracer in the foot pad, this will only be drained to this particular lymph node that only drain this anatomical area. The same, if we inject a fluorescent tracer into this foot pad, will only be delivered to this particular lymph node. And the same, if we inject in this foot pad, we will go to the axillary lymph node. And both study with fluorescent tracer with antigen delivery has been clearly shown how each lymph node specifically drain from a particular area of a particular anatomical district. And this concept is even more, I would say, explained at the level of metastasis. The lymphangiography, which are performed by surgeons to detect the sentinel lymph node, shows that a particular lymph node will drain the tumor site. Indeed, the surgeon will inject a tracer into the tumor and then follow in which lymph node this tumor drains, and this is called the sentinel lymph node. And as you know, it's particularly important because in this lymph node, if the tumor already metastasized, that's where you're likely to find metastasis. And so this concept of the sentinel node, as well as the injection of fluorescent tracer, really show and explain very clearly how each lymph node can drain from a particular anatomical area of the human body. And so to further explore this concept, we were really interested in understanding, in analyzing the efficiency of nodal filtration. Basically, we wanted to know how well a lymph node can filtrate the incoming proteome. And so to answer this question, we use two different approaches. One is to cannulate the efferent collector. So you can see a cannula here, which is ligated to an efferent collector, which will drain to a lymph node, which you don't see here because it's covered by these blood vessels. And so we can cannulate, for example, fluorochrome labeled proteins or bacteria in the pre-nodal lymph, and then collect the post-nodal lymph, and to analyze quantitatively how efficient is the lymph node in capturing this incoming proteome, which is transported by the lymph. And here you can see it's the same picture but with a fluorescent light, and you can see the cannula that is filled with fluorescence protein that directly cannulated into the lymph node. A second approach to quantify the efficiency of nodal filtration is by using quantitative proteomics. Basically, we collect the pre-nodal and the post-nodal lymph and quantified protein filtration. You can visualize all the protein that we collect in the pre-nodal lymph, and the amount of protein it's expressed in red, which means a higher amount on this scale. And these are the protein that are present in the post-nodal lymph, and the amount is green, which means lower according to this scale that we use to quantify the protein. And so you can see that in different lymph collected from different animals, there is a great rate of filtration. So proteins that are present in the pre-nodal will then filtrate it out in the post-nodal. And the Pearson coefficient, which is a statistical method that we use to see how reproducible and how good is the experiment, is very close to 1, which means that all the protein and the kind of filtration that we see in one animal, we can also see in many more animals that we tested. And these graphs here really indicated the percentage of how much protein are filtrated out from the passage through the node. So protein that are present in the pre-nodal, how much they are filtrated into the post-nodal. And this graph really showed that between 50 to 90% of protein are really filtrated out during the passage to the node. And the ones that are not, which are actually increased in the post-nodal lymph, are mostly immunoglobulin. And this result was also validated by ELISA. And both IgG and IgM increase in the post-nodal lymph, which obviously makes sense since immunoglobulin are synthesized into the node. And so to recapitulate, we performed 2 different kinds of experiments, and the ultimate goal was to understand how much can a lymph node filtrate protein, how much of the incoming proteome will be trapped basically by the lymph node. And so we use these 2 different systems. One is quantitative proteomics, so basically analyzing how much protein we have in the pre-nodal versus the post-nodal lymph, and so whatever is missing is what is captured by the lymph node. And the second approach is by cannulating a lymphatic, and you can see here the procedure in which we can cannulate. We are opening up this lymphatic, we insert the cannula, we ligate the cannula. This experiment was done in the laboratory of David Zaveja at Texas A&M. And then we can inject known amount of a fluorochrome label antigen. You can see this is a protein that is labeled in green, this is protein labeled in red, and then this is in the pre-nodal. And then we collect the post-nodal lymph, so the lymph that already trafficked through the lymph node, and we ask how much of this protein or bacteria are left over. And so just by subtracting, you can understand how is this carrying capacity of the lymph node, how much of the proteome can be really filtrated out. And here are just some quantitative measurement that shows this filtration ability of the lymph node. And you can see, for example, this is a particular protein that was injected in the pre-nodal and how much is filtrated out in the post-nodal, and the same here. And you really can see that this process, this graph shows that this process is really concentration dependent. It depends how much you inject in the pre-nodal, and you can see that a lower concentration, up to like 500 microgram, 1 milligram, the lymph node is really capable of trapping all this protein. Only when you increase around 2.4 milligram, there are some protein that start coming out into the post-nodal lymph. And this this is the same concept basically of the filtration rate in bacteria. You can see this is Staphylococcus aureus, how much we inject in the prenodal lymph and how much is left over in the postnodal. And again, you can see that it's really a process that is dependent on the amount of bacteria. If we inject 1 million or 10 million bacteria, the lymph node is really very capable in capturing these bacteria. Only when you start injecting 100 million, you can see that some bacteria start coming out into the efferent lymph. I respond to that. Yeah. So when we discuss the concept of filtration from a lymph node, what we really means is that antigen presenting cells, macrophages, dendritic cells, which are present in the lymph node, they can really capture, they can phagocytose this incoming proteome. Here you can see the dendritic cells, these are staining of a lymph node. This is an antibody that shows B cell. These are all the B cell area. And this shows the macrophages. Here is follicular dendritic cells. You can see how many of macrophages and follicular dendritic cells are present in the lymph node. There are some other population of dendritic cells which are not shown by this staining, which are also present both in the B cell area, like the follicular dendritic cells, or in the T cell area. These are mostly the common dendritic cells. And they are located in the lymph node because, again, the lymph node will filtrate the lymph that it's incoming, and these macrophages and dendritic cells have this job to basically phagocytose both self-protein coming from different tissue as well as pathogen. And these dendritic cells, upon processing of this protein, will present some of this peptide to the T cells to make them aware of which protein are entering the lymphatic circulation and the lymph node. If it's a self protein, they are making aware T cells that they should stay tolerant to this protein, whereas if it's a pathogen, they are telling the immune system basically to start an immune response to this invading pathogen. And using different techniques, we are able basically to pick up these peptides and analyzing to see which kind of proteins are constantly present by the dendritic cells to the immune system. We did several proteomic analyses, and as expected, most of these proteins derive from both intracellular and extracellular sources. And these are just a mass spec readout of several of these peptides that we were able to find in the lymph, and we were also able to elute from the MAC class 2 molecule, which is this receptor that presents this peptide to the immune system. And using different techniques, we can quantify how many of these peptides are present, so we know how many femtomole, for example, in this experiment of a certain peptide is presented from the dendritic cells to the T cells. And not only using a series of different algorithms, we can also understand basically how these peptides are generated, which proteases are important for cleaving the protein and generating these peptides. So altogether, I give you a general presentation on how the lymph is the biological fluids that collects the tissue proteome that derive from the ongoing metabolic and catabolic activity which are present in each parenchymal organ, and how the lymph nodes are very efficient in capturing and processing this incoming proteome. And really, the nodal filtration is very much dependent on the prenodal protein concentration. Also, this incoming protein will be processed by dendritic cells and macrophages, which are present in the lymph node, and they utilize a variety of processing pathways with many enzymes, which are important for generating these peptides that are then presented to the T cells for the maintenance of tolerance or for the initiation of an immune response to a pathogen. And I would like to acknowledge people in my lab that are working very hard to understand more about the lymph composition and formation and circulation, and people at UMass and Texas A&M that has been my collaborator for many years. Thank you. So I see that there is one question. How does the lymph change between pathological and physiological condition? So the lymph change very much during physiological and pathological condition. There are several proteomic analyses that have been performed by lymph collected in physiological condition, or for example during pancreatitis, during different kind of cancer, during trauma, during anthrax infection. And all these analyses really shows how the lymph capture the molecular signature of that particular organs from where it drains. And so, for example, how mediators of inflammation or mediators of tissue damage would be upregulated in the lymph collected during a trauma, for example, only few minutes after the trauma has occurred, or how different proteins which are important to fight pathogens are found in the lymph, drained from a site of inflammation very early on, and actually much earlier in the lymph than in the plasma. And so the lymph is really the biological fluid that really capture whatever is going on in every parenchymal organ, and that's why we believe in the future it could be very important for biomarker discovery during different pathogenic conditions. I don't see. Is there another question?
Yeah, it's a new one.
So yes, there is another question. How different is the lymphatic fluid draining different anatomical districts? So that's also an important question, and we have a few proteomic analyses of lymph that it drains. For example, the first one that we performed, the lymph was draining the foot. And so in our proteomic analysis, we found a lot of protein that derive from collagen, from extracellular matrix protein, because the majority of protein that are collected in that particular lymphatic district derive from the skin and the derma. That's why you find so many collagens. But other proteomic analysis that we performed later on, in which we collected the lymph, for example, from the mesenteric district, we found a lot of protein which are specific for the intestine. And so overall, the lymph collected by different districts, it is very different because it really captured the molecular signature of that particular organs. All right, so thank you very much for participating in this webinar organized by LE&RN. And again, if you have any further questions, you can just email me or get my contact information through the LE&RN organization. Thank you.